Really happy to be here. I'm sitting in my in my office today. On the left, I asked an artificial intelligence program to draw a diagram of what my laboratory works on, and in 10 seconds, that's the diagram that came up. I think it's pretty good. So what I'm going to do today is tell you about our efforts to create a molecular map of heart development, disease, and regeneration. And I'm going to talk to you specifically about two areas of investigation that are ongoing right now. Unfortunately, I can't cover everything that we're doing, but I'll try to give you a flavor of some of our interests. So let's see. These are my disclosures. So our laboratory over the past nearly 30 plus 40 years now has used muscle tissues, skeletal muscle, heart, and smooth muscle as model systems to decipher the basic mechanisms of gene regulation during development and disease. And this has really been fertile ground for discovery of fascinating molecules and mechanisms, and clearly has, I think, deep relevance to human biology, since every breath you take and every move you make ultimately comes down to the integrated function of these different types of muscles. These are some of the projects that are ongoing in our laboratory right now. I can't talk about, of course, all of these today. So I'm going to talk to you about two projects today. I'm going to talk to you about our efforts to use CRISPR gene editing to correct mutations responsible for cardiomyopathy. And then if there's time at the end, I'll tell you about our recent efforts toward reprogramming of non-cardiac cells toward a cardiac cell fate. Despite the success of lipid-lowering drugs and many other therapeutics, it's humbling to realize that heart disease remains the #1 cause of human mortality. And genetic mutations in approximately 1 out of 250 people cause devastating cardiomyopathies. So these different forms of heart disease can take many forms and have many consequences. They can include excessive hypertrophic growth of the heart, cardiac dilation and dilated cardiomyopathy, in which the heart loses pump function, or in response to acquired insults. This can caused by ischemia or other abnormalities. This can lead to loss of myocytes and replacement with fibrosis, which diminishes cardiac contractility. So over the past several years, as many of you are aware, we turned our attention to one of the most devastating, perhaps the most devastating disease of muscle, and that's Duchenne muscular dystrophy, DMD, which culminates in death in affected patients, typically in the mid-20s, due to dilated cardiomyopathy and a loss of pump function. I'm not going to talk about our work on DMD today. Many of you have heard me talk about that before, but that work really served as the springboard for the work I want to talk about, which is editing of disease genes in the heart. By way of background, we began our foray into this area of the field when we set up a muscular dystrophy clinic here in Dallas, the Wellstone Center that Jim mentioned. And patients can come in to the center. We take a blood sample and we optimize methods for correcting Duchenne, major Duchenne mutations in human iPS cells from these patients. We then built mouse models harboring the human mutations and corrected those through AAV delivery, and we're currently working on lipid nanoparticle delivery. And then we extended that work into large animals, correcting Duchenne in dogs. And this was carried out in a biotech company called Exonics Therapeutics, and has now been extended into work at Vertex in IND-enabling studies. So what I'd like to do today, though, as I said, is to focus on the genetic underpinnings of heart disease. This is a famous painting that I like from Paul Klee. Perhaps it's good that Paul Klee came along before artificial intelligence, because as you saw earlier, AI drew a picture of the heart probably equal to this in 10 seconds. So what I'm going to talk to you about today are disorders of the heart, both acquired and inherited. So we'll begin by talking about mutations in the RNA binding protein RBM20. RBM20 is a muscle-specific RNA binding protein. It's required for splicing of key muscle RNAs, for example, Titin, but also many others. Shown here on the left is a schematic of how RBM20 would function in a normal heart. The protein is synthesized, translated, of course, in the cytoplasm, and is translocated into the nucleus, where it participates in RNA granules, where it mediates splicing of the proteins required for the integrated function of the sarcomere. Mutations in RBM20 in human patients often prevent the normal nuclear localization of the protein, and this disrupts splicing and leads to dilated cardiomyopathy. The autosomal dominant mutations in a particular domain of RBM20, the RS domain, which mediates RNA binding and nuclear localization, are thought to account for somewhere between 2 and 6% of patients with dilated cardiomyopathy. This shows the amino acid sequence of the RS domain, and you can see that this region is littered with single point mutations that change amino acid codons in this region, and this disrupts the function of the RS domain. Given the severity of these mutations and their prevalence, we thought that this would be a good platform for us to test our ideas and our technologies for correcting of cardiomyopathy genes, mutations in general. Now, I think all of you are familiar with the prototypical form of gene editing first described by Jennifer Doudna and Emmanuel Charpentier, in which a Cas9 endonuclease can make a double-stranded DNA break, and this can be repaired by the repair machinery and lead to edits. And that's the approach that we took initially for correcting Duchenne mutations. Base editors developed initially by David Liu in Boston involve a Cas9 nickase, which has been incapacitated with respect to its double-stranded cutting activity, and it's fused to a deaminase domain. And this coupled protein can then be delivered to any sequence in the genome by a guide RNA, and then within a so-called editing window, this can change single nucleotides. And so in cytidine base editing, a CG nucleotide pair can be converted into a TA nucleotide pair, and in adenine base editing, an AT base pair can be converted to a GC base pair. And importantly, as I mentioned, this form of base editing does not require or involve the formation of a double-stranded DNA break. So in many respects, it's probably safer than the classical form of CRISPR-Cas9 editing. So we began by modeling RBM20 cardiomyopathy in patient-derived iPS cells. Essentially, you can convert iPS into cardiomyocytes, as all of you know. And we looked at one of the most prominent mutations. That's an R to Q arginine to glutamine mutation, which involved a single nucleotide change, a guanine to an adenine in this particular codon. And we thought that this lent itself ideally to adenine base editing as a initial test case. So we screened a variety of guide RNAs, and we ultimately settled on the one shown here in blue, which has a so-called PAM sequence that directs the Cas9 to its target. And you can see the target adenine here, and here's a bystander, so-called bystander adenine at position 14 within the guide RNA. For the aficionados on the call, we tested a variety of base editors, and we ultimately settled on ABE Max with a VRQR SPCas9 modified PAM recognition sequence. And so testing this in these human cells in vitro, we found that this particular guide RNA and this base editor had remarkable efficacy in editing. Here at the target nucleotide at position 6, it edited at about 90% efficiency, and there was no bystander editing at this adjacent adenine. So we moved forward with this and looked at its consequences on cardiac pathology. So this shows human cardiomyocytes in vitro. You can see the well-organized sarcomeres, and you can see RBM20 in green in the nuclear puncti where it mediates splicing. And here you can see in the homozygous mutant cells, now RBM20, due to that single point mutation, is prevented from appropriate translocation to the nucleus, and the sarcomeres are disrupted as a consequence of defective splicing of its targets. And following A-base editing, really was quite remarkable in these early experiments that the sarcomere integrity was faithfully restored, and RBM20 was restored to its rightful location in the nucleus. So we went on then to model this in vivo, and other groups have done this as well. Lars Steinmetz at Stanford and EMBL has also done beautiful work in this area. So here we made RBM20 mutant mice, and here you can see the homozygous mutant mice have severely enlarged hearts with a dramatically diminished fractional shortening, indicative of cardiomyopathy. We then tested whether we could correct this mutation in vivo. The base editor is too large to be incorporated into a single adeno-associated virus, AAV. So we used a split system where we could put the base editor fused to the Cas9 nickase with a protein intron, a so-called N-intine, at one end, and then we put the other half of the base editor in a second vector with the C-intine, and when this is introduced into cells, it mediates protein splicing and reconstitutes the functional base editor. And this was placed under control of the cardiac-specific troponin T promoter to restrict its expression following systemic delivery to the heart. And we used 2 guide RNA cassettes in each vector to deliver the base editor to its target. And so the initial strategy that we used was we injected 1.25 times 10 to the 14 viral genomes per kg of the virus intraperitoneally into these mice at an early age prior to the severe onset of cardiomyopathy. And here's the results that we obtained. So at 12 weeks of age, you can see the homozygous mutant mice have really severe cardiac disease with dilation of the left ventricle, and the corrected hearts were virtually indistinguishable at a histological or functional level from wild type. And here you can see the lethality associated with the homozygous mutant, and lethality is completely blocked by systemic editing with this base editor. This shows 4 replicates of hearts in which we carried out extensive RNA sequencing. You can see the really dramatic dysregulation of cardiac gene expression in the mutants, and you can see how the genes that are downregulated and the genes that are upregulated in the mutant hearts are largely restored to normal expression in the corrected hearts. And here you can see just an example of one of those. All of you know that NPPA, ANF is a hallmark of cardiac stress. It's upregulated here in the mutant, and in the corrected, it's brought back down to baseline. And I told you that titin is probably the most important target for splicing by RBM20. And here you can see that the adult isoform of titin is expressed in the heart. In the mutant, the adult isoform is extinguished, and upon correction, the adult isoform is returned to normal levels of expression. Here you can see the histological consequences of this mutation and its correction. So here you can see this are from the hearts of these mice. Here you can see in the mutant hearts, RBM20 is restricted to the cytoplasm, and it exists in puncti here outside the nucleus. And then following correction, it's really quite remarkable. I think that RBM20 is so faithfully returned to its nuclear localization, and the sarcomere organization is restored. So based on this, we really feel that base editing, modulation of a single nucleotide, has a future for correction of catastrophic cardiomyopathy mutations, such as those in RBM20. And we're continuing to extend this work through alternative delivery strategies, as well as a newer technology referred to as prime editing, which I won't go into today, but prime editing makes it possible to correct large stretches of nucleotides with a single guide RNA. And I think that really opens up a lot of potential opportunities. So I'd like to turn now to hypertrophic cardiomyopathy. And the classic hypertrophic cardiomyopathy is caused by mutations in myosin heavy chain 7. So this, in contrast to dilated cardiomyopathy, which results in loss of myocytes and chamber dilation, hypertrophic cardiomyopathy takes the form of diminished chamber size and pathologically thickened ventricular walls, as you can see schematically here. Many hypertrophic cardiomyopathy patients harbor a mutation in the dominant myosin of the human heart, MYH7, which participates in the sarcomere as schematized here. Approximately 1 in 3,000 people harbors a myosin heavy chain 7 mutation, and about 2% of these harbor the arginine 403 to glutamine mutation, which was the first mutation discovered and characterized in humans by the Seidman lab. And this is an extremely severe mutation which causes hypercontractility, hypertrophy, and cardiac sudden death. So about half the patients with this mutation die of cardiac sudden death by the age of 45. And this mutation essentially diminishes, reduces myosin's ability to be released during contraction and leads to hypertrophy. So we looked at this region, the ARG403 region, and the mutation that causes this disease is a G to an A mutation, as I mentioned. And so we screened a variety of base editors. In this case, we screened 8 different SPCas9 variants with different PAM requirements, and we screened a variety of guide RNAs. We ultimately settled on one guide RNA called human 403 SG RNA, because we realized that if we could edit this nucleotide, we would convert pathogenic glutamine back to an arginine. And I'll note that there are additional adenines within the guide RNA region. And so we did have concerns that they could lead to bystander edits, and it was important to document the specificity of this guide RNA for this form of editing. And so this experiment shows that. So here we tested our optimized guide RNA. Again, for the aficionados in the audience, the optimized guide RNA was ABE max SPCas9 with an NG PAM and an optimized guide. And here you can see these are in 2 different patients. These are human patient heterozygous for the mutation, that this editing efficiency borders on 100% efficient. And even at the bystander edit, adenine 11, the bystander editing is well below even 1%. And there's essentially no off-target or bystander editing at the other adenine. So this looked to be a really effective base editor for the human genome, at least based on these in vitro experiments. So whereas myosin heavy chain 7 is the dominant myosin, as most of the audience well knows in humans, in mice, the dominant myosin is MYH6, also known as alpha myosin heavy chain, versus beta myosin heavy chain in humans. Now, these proteins are highly homologous, and particularly within the region of the glutamine mutation, the amino acid sequences are identical between the mouse and the human, but the nucleotide sequences are different, as indicated by the absence of asterisks at the mismatched nucleotides. So in order to test whether the human guide that we had optimized in human iPS cells would work in vivo, we needed to make humanized mice. And so we generated mice in which we introduced 75 nucleotides spanning this region of the mutation. we introduced those into humanized mice, and then we could test our human guides in this mouse model. So here you can see the heterozygous myosin heavy chain 6 mutation in mice. It leads to severe hypertrophy. You can see that here. Here you can see fibrosis in these diseased hearts detected by picoseries red staining, and you can see myocyte disarray. So we thought this was a useful model to test whether we could correct this mutation in vivo, and this was done by Andreas Chai, an MD PhD student in our lab. So I won't go into all this data. It was published very recently. Here you can see a few markers of cardiac disease. Wall thickness is increased. That's brought back to normal by base editing in this mutant mouse line. Heart weight to tibia length is normalized. Collagen area indicative of fibrosis is normalized. And sensitive stress marker, NPPA, is brought back down to baseline by base editing in vivo. So we feel that this work really highlights the potential of gene editing strategies for correction of hypertrophic cardiomyopathy mutations. And I'll say that the Seidman group published a paper back-to-back with ours earlier just last year, describing similar results. Now, although genetic mutations are a common cause of heart disease, as I've mentioned, estimated to exist in 1 in 250 people, many of these mutations are obviously different, and it becomes a challenge to generate optimized guide RNAs for every single type of point mutation. And so we wanted to ask whether we could expand the potential of this technology to the most common forms of heart disease, and those are acquired forms of heart disease, most commonly caused by ischemia, as occurs in coronary artery disease, leading to infarction. And so the question we ask specifically about this form of heart disease, but really more broadly with respect to virtually any disease that you can think of that might be caused by pathological activation of a signaling pathway, was whether it might be possible to deploy base editing to modify single amino acid target sequences in effector proteins that lead to pathological responses and death or dysfunction of target tissues. And as you all know, there are myriad post-translational modifications of specific amino acids that might lend themselves to this form of gene editing, which we thought, at least in principle, could disrupt the pathogenic activation of a pathway, but leave the normal physiological actions of a pathway unperturbed. So over almost 2 decades now, our lab has been interested in signaling by the calcium calmodulin dependent protein kinase, CAMK2. This work was initiated by Johannes Bachs when he was in our laboratory, and many other people have worked on the role of CAM kinase signaling in cardiac pathology, notably Mark Anderson's group, Joan Heller-Brown, and Lars Meyer, among others. Our work and others showed that when CAMK2 delta, the dominant CAMK isoform in the heart, is activated by ischemia, by neurohumoral signaling, and other stresses, that it leads to dysregulation of contraction, calcium cycling, and importantly, transcription, where we and others showed that CAMK2 is the key trigger for pathologic transcription through its repressive activity of the histone deacetylase HDAC4. So this was the proof of concept pathway that we wanted to test our ideas on. And ultimately, this pathway, when pathologically activated, as most or all of you know, leads to many pathological downstream consequences in the heart. So CAMK2 delta has 3 domains. It has a catalytic domain, a regulatory domain, and an association domain. In the unstressed state, the catalytic domain is sequestered by the regulatory domain, which holds its activity in check. But in response to various forms of stress, the regulatory domain can be post-translationally modified. In response to ischemia, as shown by Mark Anderson initially, two adjacent methionines in the regulatory domain become oxidized, and this unleashes the catalytic domain to uncontrollably phosphorylate downstream targets, leading to pathologic cardiac remodeling. In addition, threonine 287 in the regulatory domain can also be pathologically phosphorylated, leading to unrestrained activity of the catalytic domain. So Simon Lebeck, a German cardiologist who came to our lab with the idea of using base editing to modulate CAMK activity, looked carefully at this region of CAMK2. And as all of you well know, methionines, the two targets for pathologic oxidation in the regulatory domain, are encoded by ATG codons. And so we wondered whether those ATG codons would lend themselves to adenine base editing to change the amino acid sequence and thereby render CAMK2 resistant to pathologic activation. So we screened a variety of base editors. I won't go into all the details. For the experts in the audience, I'll simply say that we ultimately settled on ABE8E SPRY, which has a specific editing window, and we identified the optimum guide RNA to base edit these target sequences. The target sequences for editing in this case were adenine 17 and 14, but we noted that there were adjacent adenines which we had some concerns could lead to bystander editing. So here you can see the efficiency and the specificity of editing of those target sequences. This is in human cells. So A17 and 14, the two targets for converting the methionines into valines, were edited at least 80% efficiency. There was one bystander edit at A10, which would convert a histidine to an arginine, and we don't know the functional consequence of that particular bystander edit, although we have not seen any evidence that it has pathogenic consequences. So we initially then, using those edited human cardiomyocytes, we tested whether they would be resistant to CAMK pathogenic activation in response to ischemia, reoxygenation, injury. We used this paradigm, a well-established paradigm in vitro, to subject human cardiomyocytes to ischemia followed by oxygenation, reoxygenation. Here you can see CAMK oxidation at those two critical methionines in wild type, and here you can see the edited CAMK does not undergo oxidation of those sites. And here you can see the activation of CAMK2 in response to the activation of the enzyme in response to oxidation of those methionines, and that's diminished in the mutant. So we were confident, at least from these in vitro studies, that blockade to oxidation of those two methionines could have functional significance. So we tested this in vivo. The experimental paradigm we used is shown here. I won't go into all the details. This work was very recently, some of this work was very recently published. Suffice it to say, we subjected mice to ischemia for 45 minutes, followed by reperfusion, which has been shown to lead to oxidation of those two critical methionines and hyperactivation of CAMK2. And then we monitored cardiac function at several weeks thereafter. This shows the specificity following injection. In this case, we used AAV9 again to inject the base editing components directly into the ischemic zone. In this case, there's several advantages to this approach. One is you're injecting the virus directly into the cardiac lesion, so it's highly specific, and the dose of the virus is far lower than as would occur in a systemic delivery. And here you can see the anatomical restriction of the editing of these target adenines in the anterior wall at the site of injection, and there's virtually no leakage to distal regions of the heart. Here you can see the consequences of this approach on cardiac function measured by echocardiography. In sham, the ejection fractional shortening hovers around 58%. There's a dramatic diminution in cardiac function following IR injury, and in the edited mice, it's restored to normal. And I think this really shows it visually. This is a transverse section through the heart. Here you can see the normal heart with the LV. Here's the ischemic zone stained with picoserous red for fibrosis, and here's the edited mice that you can see are corrected. So we also saw, and I won't go into all the data since some of it's published, but we also saw that there was a transient decline in cardiac function within 24 hours following ischemia reperfusion, and the edited mice then gradually over the ensuing 2 weeks returned to the level of fractional shortening that I showed you in the previous echo cardiogram. So to summarize this work, which was very recently published, we want, well, to extend this work, I'll say, we wanted to extend this work from the mouse to ask, could we really translate this toward the human setting? And so in this case, we created humanized mice, once again, where we took the regulatory region that's subject to these post-translational modifications, and we inserted this region encoded by exons 11 and 12 for the aficionados of CAM-K in the audience. We replaced the mouse gene with the human gene, so now we have mice that harbor the target genomic sequence from human, and we can then use human guides to edit those target nucleotides. And I won't go through all the data. You can see it here, and some of it was just very recently published. In this case, we used a version of AAV called MyoAAV with the A base editor at a very low dose of the virus. And you can see that it is able to sustain cardiac function following ischemia reperfusion injury. One of the questions that we had, reviewers had also, was what happens to the exercise tolerance of mice that have been subjected to CAM-K editing? And I can tell you that we have not seen any impact on exercise tolerance, nor have we seen any long-term consequences of editing of those amino acids during the entire lifespan of mice. So what I've told you then so far is that these are the two target methionines encoded by ATG codons in the regulatory domain. In response to ischemia reperfusion injury and oxidative stress, they become oxidized, and this leads to pathological CAM-K activation, which culminates in impaired contractility, fibrosis, and pathological cardiac remodeling. And as a proof of concept, I've told you that base editing of two adenines to guanines changes the target amino acids from methionine to valine. And this does not affect the physiologic, as far as we can tell, the physiologic function of CAM-K2, but it impairs the pathologic activation and then renders the heart resistant to this form of stress-induced cardiac disease. So this is, again, the regulatory domain. These are the two target methionines that I just showed you that we had base edited to impose a blockade to pathologic activation. There's another modification of this regulatory domain, and that's at threonine 287, which becomes hyperphosphorylated and also can unleash the activity of the catalytic domain. So Simon Lebeck also looked at the possibility of editing that threonine through the same sort of logic that we applied to the methionine edits. Here's the target sequence. Here's the threonine encoded by an ACT codon. So again, that lent itself, we believed, to A base editing. And so by converting this adenine to guanine, it would in principle convert threonine into alanine and render the enzyme insensitive to hyperphosphorylation at that site. So and that was indeed the case. In this case, because phosphorylation of that site had been implicated in prior studies by others, that in severe thoracic aortic constriction, so-called STAC, which is a model for severe pressure overload, that target threonine becomes autophosphorylated by CAM-K. So this is the model that we tested. We took mice, subjected them at adulthood to STAC, and then we monitored their cardiac function by echo, et cetera. So here you see a survival curve. STAC is a very severe form of cardiac stress. It leads to severe lethality, in fact, with more than half the mice dying within 2 weeks after this form of constriction. And the lethality in response to this was almost abolished in these edited mice. So we looked more deeply. Here's the autophosphorylation of threonine 287 as detected with an antibody against that phosphorylated residue of CAMK2. And here you can see then in the threonine 287 to A base edited mice that hyperphosphorylation of the target threonine 287 is prevented. And here you can see a quantification of CAM kinase activity measured in cardiac extracts is diminished in response to a base editing of that single nucleotide within that codon. Here you can see the consequences on cardiac function. Here's wild type echo, here's STAC, really dramatic diminution in cardiac contractility. Here's sham, and here's the edited mice. They're not brought back entirely to normal, but they really do show remarkable resistance to the impact of STAC. And here's some histology. Here's the integrated architecture of cardiomyocytes in wild type mice subjected to sham operation. Here's the pressure overloaded STAC mice, which have ventricular dilation and cardiac fibrosis. And here's the edited mice editing the single nucleotide, converting the threonine 287 to A. Fibrosis is prevented and architecture is sustained, normal architecture is sustained. And here you can see just as one measure of cardiac pathology, cardiac apoptosis is diminished by blockade of phosphorylation of that critical site. So this is really, I think, a dramatic demonstration of the impact of a single base edit of a one codon in this critical signaling molecule on the gene expression in the heart. So here in triplicate, you can see changes in gene expression. These genes are downregulated in response to STAC. That downregulation is blocked in the threonine 287 edited mice. These genes are upregulated in wild-type mice subjected to STAC, and the upregulation of those genes is diminished by editing of the single nucleotide. And here are the gene families that respond to this single amino acid change in this critical signaling molecule of CAMK2 delta. So cardiac function genes, which are downregulated in response to cardiac stress, their expression is preserved upon editing. And here's the cardiac disease genes that are upregulated in response to stress, many of them encoding fibrotic components and inflammatory response genes, and those are brought back to baseline by editing at this site. So what I've told you then is a second example, and this I think just came out maybe this week or so, or maybe last week. I've told you that editing of an autophosphorylation site, a threonine within the regulatory domain, converting this to an alanine can render CAMK2 resistant to hyperactivation, pathologic phosphorylation that leads to cardiac disease, and in most other respects, as far as we can tell, these hearts retain normal function. So there are several issues that we're currently considering with respect to our gene editing studies, both with Duchenne and with cardiomyopathy mutations, as I've described. Those are what is the spectrum of correctable mutations, and what are the new disease genes, new diseases that might be susceptible to these forms of therapeutic intervention? When in a disease process might it be best to intervene? Certainly the earlier the better, but that may vary depending on the particular disease and the particular stress. What is the optimum mode of delivery? Viral is the approach that we've taken so far because it's the most efficient, but I think many of us would agree that the long-term goal in this field would be to deploy non-viral delivery systems, perhaps lipid nanoparticles, as were pioneered during the COVID era for the COVID vaccines. How important is it for immunosuppression in these settings? That's a really important question. And what are the strategies for getting rid of Cas9 so that an endonuclease with gene editing potential is not existing in the target cells for the lifetime of the patient? And those are some of the issues that we're addressing. In the last few minutes, I'd like to shift gears and talk to you about an alternative strategy to restore cardiac function in the setting of cardiac coronary artery ligation and infarction. So one of the long-term goals in this field by us and many others has been to reprogram cells, fibroblasts that would otherwise form a scar into functional cardiac myocytes, so-called induced cardiac myocytes, by injecting reprogramming factors or combinations of drugs into the infarct zone. And so several years ago, we carried out a screen to identify epigenetic and novel factors that could promote reprogramming of fibroblasts into cardiomyocytes. Now, this field of cardiac reprogramming has been really pioneered by Deepak Srivastava, a former member of our laboratory, and many other labs, particularly Lee Kwan and Masaka Aida and others. The approach we took was to optimize a recipe for reprogramming, which was a 5-component cocktail that's shown here, and this work's been published. We abbreviate this AGHMT. We then screened through 1,500 transcription factors and epigenetic factors in retroviruses for any of those that might enhance the reprogramming process in the presence of these factors. And one factor, which frankly we were unfamiliar with, is known as PHF7. And you can see here in the reprogramming assay, looking at expression of troponin T or alpha myosin heavy chain as a marker, it really stood out from the crowd by its ability to augment the reprogramming activity of these factors. PHF7 is a little studied protein. It's an E3 ligase and a histone reader. It belongs to the plant homeodomain finger family. It contains two homeodomains and a ring finger domain, and it binds histones H2A, H3, K4 dimethyl. So it's an epigenetic regulator that can identify specific modifications or targets in the genome and modulate their activity. We showed that PHF7 in work that was published is expressed in MESP1 positive cardiac progenitors during development, but is not expressed notably in the adult heart. So this is one example in which we tested the effects of PHF7 on cardiac reprogramming, again, in the presence of our initially optimum cardiac cocktail of 5-component cocktail, shown here as AGHMT. And one of the things that stood out to us early on was that in the presence of PHF7, the reprogramming of the most resistant type of cell that we or others had identified, the adult fibroblasts, was accelerated. So typically, it takes about 7 days for adult fibroblasts in the presence of various reprogramming factors to begin to acquire a cardiac fate. But when PHF7 was added to the mix, this was accelerated such that even at 1 day after addition of PHF7, here you can see on a FACS plot, 20% of the fibroblasts were converted into a cardiac phenotype as marked by cardiac troponin T and alpha MHC GFP. And here you can see the consequences. These are single wells of a plate. And here you can see quite dramatically, if you just look at cardiac troponin T, in the presence of what was initially our optimum cocktail, and then the addition of PHF7 really gives it a dramatic boost. So PHF7 also it dramatically enhances adult human cardiac fibroblasts. You can see that here with alpha-actinin as a marker. And here you can see the effects of PHF7 on top of the previously optimized, one of the previously optimized cocktails. In humans, the cocktail is a little different than in mice. In humans, the cocktail is myocardin plus GATA4, HAND2, MEF2, and TBX. And then, so in the presence of this cocktail, you can see that PHF7 really boosts the expression of these representative cardiac genes. So here's yet another example. We looked at the consequences of PHF7 expression in, these are in mouse tail-tip fibroblasts in the presence of this optimum cocktail. And you can see that the addition of PHF7 quite dramatically diminishes the expression of a large number of genes and augments the expression of a large number of genes. And if you look at what these genes are, so PHF7 suppresses many genes involved in inflammation and other pathogenic responses, and you can see that it strongly enhances a number of cardiac genes. So we think that PHF7 somehow is potentiating the cardiogenic activity of these core transcription factors in the reprogramming cocktail. So how does PHF7 do this? And I don't have all the answers to that question, but I'll tell you about our efforts to begin to try to decipher some answers. So here we carried out reprogramming in mouse embryonic fibroblasts, and then we looked at their conversion to induced cardiac-like myocytes with the canonical reprogramming cocktail in the presence or absence of PHF7. And then we did chromatin IP to ask, where does PHF7 go within the genome? And this was quite interesting. I won't go through all the details because of time and because it's a complex slide. But here you can see that in the presence of the core cardiac transcription factors, PHF7 is directed, this is in a chip assay, it's directed to the target genes in the presence of these factors, whereas in the absence of these factors, it localizes to a different subset of genes. And we ask, what are the nucleotide sequences embedded in these target sequences that are to which PHF7 is recruited? And this was quite interesting. If you look at the sequences beneath the PHF7 ChIP-seq peaks, you can see that they are predominantly occupied by key transcription factors involved in the reprogramming process. And the downstream target genes associated with these ChIP-seq peaks, you can see here, are also enriched in cardiac genes. So to summarize a lot of work, this was work initiated by Huanyu Zhu when he was in the lab and extended by Glynis Gary, a cardiology fellow in our lab. These were the conclusions of that initial in vitro study, and that was that PHF7 can overcome, we believe, barriers to reprogramming and stabilize transcription factor binding at cardiac enhancers. We know from prior work that PHF7 recognizes and binds H3K4 dimethyl marks, which are well known from work of others to identify primed enhancers. And in work which we've published, so I won't go into it in detail, we've shown by proteomics that PHF7 cooperates with the SWI/SNF complex and with SMARCD3 to increase binding and accessibility of cardiac transcription factors to super enhancers. And I showed you some of the chromatin IP data to support that. Now, in the last few minutes, I'll just tell you about our initial unpublished work in which we've begun to ask, can PHF7 really promote reprogramming of fibroblasts, which would otherwise form a scar? Can it reprogram them into cardiomyocytes in an in vivo infarcted heart? So to test this, we performed LAD ligation in 10 to 12-week-old mice, followed by a single intramyocardial injection of retrovirus containing expression vectors for each of these factors abbreviated as shown here. We then performed echo at 24 hours post operation and then assessed injury at 1 and 3 weeks and 16 weeks post MI. So when we perform these experiments following coronary artery ligation, there's a precipitous decline in cardiac function measured by ejection fraction. And if you inject with GFP as a control, that remains diminished. If you inject the PT cocktail, that's PHF7 plus TBX5, you get an improvement in function. And in all combinations of cocktails in the presence of PHF7, you get improved cardiac function. The strongest in vivo combination shown by Deepak and others for reprogramming is GMT, and we believe that PHF7 can replace components of this cocktail as shown by the comparable restoration of ejection fraction with the PT versus the GMT or other cocktails. This shows the consequences on muscularity and fibrosis following injection of each of these different cocktails. I won't go into all the details due to time, but you can see that each of these different cocktails can diminish fibrosis to varying extents and can enhance muscularity at the infarct region. So lastly, then, we wanted to try to perform lineage tracing to ask where does PHF7 act and how does it act in the infarct region of these hearts. And to do this, we were able to use the genetic strategy that was pioneered by Jeff Mulcantine and his lab, in which they introduced the Mercury-Mer cassette within the periostin gene, periostin being a highly sensitive marker of fibrotic stress in the infarcted heart. And we then used a strategy with tamoxifen treatment where we could activate a tomato reporter under control of the periostin Mercury-Mer gene, and we tested this in these hearts. So this shows the region of the infarct. This is a bright field image looking at tomato. So tomato's been activated here in this Mercury-Mer system from Mulcantine, and this is 21 days post-MI. You can see in the infarct region that the fibrotic pathologic activation is indicative of this disease region of the heart. So here we've looked at the various PHF7 cocktails. I won't go through all the details. Suffice it to say that when we do a merge of the tomato marker, which is, again, the marker of the fibroblast lineage, and we look for its overlap with cardiac troponin T in orange, you can see there's a quite significant overlap, suggesting that cells in which the fibrotic marker were activated may have adopted a cardiac fate or contributed to the formation of cardiomyocytes marked by cardiac troponin T. So lastly, we've asked, well, what can PHF7 do on its own, if anything? And this is an ongoing preliminary work, which we're still trying to understand. But as we've shown here, when we inject retrovirus encoding simply PHF7 alone into the infarct zone, it can improve cardiac function, and it can improve muscularity of the heart and diminish cardiac fibrosis. And you can see that here by immunostaining. So the last 2 slides, I'll show you some ongoing work that is still in progress. We're trying to ask, at the single nucleus level, in infarcted hearts that have been injected with reprogramming factors, what is the consequence of expression of PHF7 or other reprogramming factors shown here on the fate of the cells in which they are expressed? And so to do that, we carried out, as I said, single nuclear RNA sequencing, and this generates a UMAP plot, as you can see here. And within this UMAP plot, based on markers of each of these cell types, there's a dozen or more than a dozen different cell types. And in particular, I'll call your attention today to 2 populations. There's a fibroblast set of population shown here, a cardiomyocyte population here with diverse types of cardiomyocytes. And that's consistent with work from Miao Cui in our lab that identifying various subtypes of cardiomyocytes in the injured or regenerating heart. So here we've split out these UMAP plots. This is in a GFP control injected heart, and here's in a PHF7 injected heart. And what you can note in the PHF7 injected heart, the fibroblast population here is diminished, and the cardiomyocyte population seems to be enhanced. And when we superimpose these populations on top of the periostin induced tomato marker, you can see that what appears to be the reorientation, I'll say, of fibroblasts marked by TD tomato from the periostin reporter toward a cardiac fate. And we're continuing to try to understand how this works and what is the trajectory and origin of these various cell types as indicated here in these single nuclear plots. So what I've told you then is in my last slide that PHF7 is an epigenetic reader that can be directed to cardiac promoters, presumably through its interaction with histone marks and with cardiac factors themselves. And we're currently exploring the mechanistic basis of PHF7 reprogramming and currently looking at non-viral delivery strategies for introduction of these reprogramming factors into infarcted hearts. So this brings me back to my first slide. These are the projects, again, that are currently being pursued in our lab. I've talked to you today about our efforts to use CRISPR correction of cardiomyopathy and to begin to explore the role of epigenetic factors in cardiac reprogramming. If you ever invite me back again, I'll hopefully have time to talk about some of these other projects that are, at least to me, equally exciting. So I'm very fortunate to have been part of just an incredible legacy of students and postdocs in my lab, many of whom are on this call. The people that were involved in this work, I won't have time to go through all their names individually for the sake of time. We've had a great myo-editing team. Many of these people have moved on to establish their own labs in academia or to extend our work on Duchenne at Vertex. The work on cardiac reprogramming was carried out by Glynis Gary and these other individuals. We've had amazing collaborators and technical support in our lab. These are our sources of support. And all the work in my lab is carried out as a collaborative effort, not only with our team, but with my two partners, Rhonda Basil-Duby and Ning Liu, who without them, I would not be able to be speaking to you about this work today. So I want to express my gratitude to the entire team and to them in particular. And I'm happy to take any questions. So let's stop share.